Assalamu alaikum. Today my topic is growth incubator, also known as thermostat incubator. A growth incubator used in laboratories, often referred to as a microbiological or CO2 incubator, serves a specific purpose in facilitating the growth of microorganisms or cell cultures under controlled conditions. Laboratory growth incubators provide a controlled environment for the growth and cultivation of microorganisms, cell cultures, tissues, and other biological samples. They maintain specific conditions such as temperature, humidity, and gas composition to support optimal growth. Most growth incubators allow precise temperature control within a range typically between ambient temperature and 50 degrees Celsius, depending on the requirements of cultures being grown. Some models offer both heat and cooling capabilities for a wider range of applications. Certain cell cultures and microorganisms require specific humidity levels for optimal growth. Growth incubators often feature humidity control systems to maintain the desired moisture content within the incubation chamber. Some incubators provide the option to control the atmospheric composition within the chamber, particularly the concentration of gases such as CO2 and oxygen. This feature is crucial for culturing cells that require specific gas concentrations for growth, such as mammalian cells or anaerobic microorganisms. To prevent contamination of cultures, growth incubators are equipped with sterilization features such as high temperature sterilization cycles or UV irradiation. These processes help maintain a sterile environment within the chamber. Advanced growth incubators are equipped with sensors to monitor temperature, humidity, gas concentrations, and other parameters. They also have alarm systems to alert users in case of deviations from the set conditions, helping to prevent damage to cultures. Growth incubators find applications in various fields, including microbiology, cell biology, tissue culture, pharmaceutical research, biotechnology, and clinical diagnostics. They are essential for maintaining cultures for experiments, production, and testing purposes. Growth incubators are typically designed to be compact with airtight chambers made of corrosion-resistant materials such as stainless steel. They often feature shelves or racks for easy placement of culture vessels and adjustable airflow systems to ensure uniform distribution of temperature. Regular maintenance, calibration, and validation are essential to ensure the accuracy and reliability of growth incubators' performance. Manufacturers often provide guidelines and services for proper maintenance and calibration of their incubator models. A thermostat incubator is often referred to as a growth incubator. These terms are sometimes used interchangeably, especially in the context of laboratory equipment designed to provide controlled conditions for the growth of microorganisms or cells. While the term thermostat incubator emphasizes the temperature control aspect, the term growth incubator may encompass a broader range of features and functions, including humidity control, gas composition control such as CO2 and O2 levels, and sterility maintenance, depending on the specific requirements of the cultures being grown. PV stands for process variable and SV stands for set point variable. These terms are commonly used in control systems and automation to describe the variables involved in maintaining a desired condition for set point within a system, such as temperature, humidity, or gas concentrations in the case of a growth incubator. The process variable refers to the current value of the condition being controlled or monitored within the system. For example, in a growth incubator, the PV could represent the actual temperature, humidity, or gas concentration measured inside the incubation chamber at any given time. Set point variable is the target value or desired condition that the system aims to achieve and maintain. In a growth incubator, the SV would be the preset temperature, humidity level, or gas concentration that the user specifies as optimal for the growth of the particular microorganisms or cell being cultured. Petri dishes are placed in a growth incubator to provide a controlled environment that promotes the optimal growth and development of microorganisms or cell cultures. Growth incubators maintain a stable temperature conducive to the growth of the specific microorganisms or cells being cultured. Incubators provide a controlled, sterile environment necessary for culturing microorganisms. By placing petri dishes in the incubator, the risk of contamination from airborne particles or other sources is minimized. Incubators help maintain the appropriate humidity levels within the petri dishes. Adequate moisture is essential for the growth of microorganisms and prevent the drying out of agar-based culture media. Some microorganisms, particularly those used in cell culture, require specific atmospheric conditions, such as controlled levels of carbon dioxide. Growth incubators can regulate the gas composition within the chamber to meet these requirements. Placing petri dishes in the incubator protects the cultures from external disturbance and provides a stable environment for observing and monitoring. By incubating petri dishes in a controlled environment, researchers can ensure that the conditions are optimized for the growth of the specific microorganisms or cells they are studying. This enhances reproducibility and reliability of experimental results. Placing petri dishes in a growth incubator ensures that the cultures receive the necessary conditions for robust growth, enabling researchers to conduct experiments, perform tests, and observe microbial behavior under controlled conditions.